Hey guys, today we're taking notes on DNA during the cell cycle. So here, um, because our DNA takes on different forms throughout the cell cycle, it's going to have different names. So depending on what state or what form it's taking, it's going to be called something different. So biology is also known as a language sometimes because there's so many different vocab words. And sometimes the words mean something very similar, but there's just little nuances that make the words different. And so this is one of the scenarios where there's lots of vocab and they mean similar things, but they're just a little bit different. And what DNA actually is it's simply a set of instructions on how to make proteins in your cells. And really, it's the proteins in your cells that give you your traits. They're the ones that do the jobs in your body that that let materials into your cell or out of your cell. They determine the color, your DNA determines the color of your eyes, or all of the traits about you. But your DNA is a set of instructions on how to make proteins, and proteins do the jobs that make your eyes a certain color, or that make your nails grow a certain speed, or if you determine how well you can smell or see or anything like that. So it's all in your DNA, and your DNA is simply a set of instructions on how to make proteins. Proteins are a big deal. They do lots of stuff in our cells. And this right here is a karyotype. It is a picture of human chromosomes or human strands of DNA. We have 46 chromosomes or 46 strands of DNA in each of the nuclei in our cells. And so we think of it like this. We have 23 pairs of chromosomes. So you have two first chromosomes. You have one from your mom and one from your dad. You have two second chromosomes, one from your mom, one from your dad, so on and so forth. So here we have 23 sets of chromosomes, 23 from mom and 23 from dad. 1 through 22, these are called autosomes. And right here, the X and the Y, these are called sex chromosomes. These show us whether or not the person is a male or a female. And this person has a very short little Y chromosome, so that means that this is a karyotype of a male. Because if you have a Y, you're a guy. And if you're a girl, you have two X chromosomes. So here, we're going to start talking about the different forms that your DNA or your chromosomes take during the cell cycle. So let's start with interphase. Since interphase is considered the normal cell life, let's start there. We know that interphase is made up of three phases, G1, S, G2. And so this is normal cell life. And during normal cell life, we need to make proteins. And in order to make proteins, we need to be able to read the DNA. So the DNA is opened up. Let's think of a newspaper. It's opened up so it can be accessed and it can be read, so that proteins can be made. Later on during the M phase, we're going to sort of crinkle up this, or coil it, and we're not going to be able to access it to be able to make proteins. But right now, during G1, S, and G2, it's very important that we are able to read the DNA. So remember, in G1, we have to make proteins in order to get ready for synthesis or DNA replication. And we know that during G2, we need to make proteins because we're going to be dividing soon and we need to get ready for mitosis. So it's very important that the DNA is in this relaxed, uncoiled state. And we call this relaxed, uncoiled state, the DNA or the chromosomes are in chromatin. They are in chromatin form. And so what I want you to do is think of like a bowl of spaghetti. All of those chromosomes are just relaxed and uncoiled. And here, if you look at this picture up here, we can see that, let me circle one of these. Let me get my draw tool. So here you can't really see, oh, let me draw again. Here we go. You can't see the individual chromosomes here. You can just sort of see a blob in the nucleus. And that's because at this point, let me turn my draw tool off. That's because at this point, the individual strands of DNA are too thin. So think of it as like thread. If you stand far away from this spool of thread, you can't really see each individual line of thread, but you can see the big spool of thread. And that's because the thread is too thin, but you can see it all when it's all together. You can see the blob. And that's the same is true here with the DNA during interphase. It's very, very small, and it's uncoiled, so it's nice and relaxed, and you can't really see individual strands. Later on, let me circle another one. We'll be able to see how they coil up, and you can see individual chromosomes. But that's later on. That's not now. So let's uncircle that. All right. Here, now we're going to be talking about the G1. So here in G1, we have 46 chromosomes. And remember that they're in the chromatin state. They're relaxed. And so here we have 46 chromosomes. Let me draw this out. 46 chromosomes. And we have, so let's just draw this one line right here. Our DNA looks like that. It's a line, just like that. And then during S phase, we know that our chromosomes are going to be replicated. So if we had 46 chromosomes like that before during G1, then we're going to copy it. So there's one here, and then here's its exact copy. So that when we get into G2 phase, let me scoot over here, we have interphase, we have 46 of these. Then we go into S phase, and we have 46 chromosomes, and they are attached to their identical copy that was made during S phase. So we have 46 chromosomes and their identical copies. And we call these chromosomes that are attached, these identical copies. So here is here's one copy of it, and then here's the identical copy on the other side. So we call these sister chromatids, and they're attached right in the middle here. We call these sister chromatids, and they're identical copies. And so we have 46 chromosomes and their identical copies. So if you were to think about it, don't get confused here, but if you really want to think about it, we would have 93 individual chromatids. That means that we have 46 of these chromosomes and then the identical copies. But if that confuses you, don't think too hard about it. Don't worry about it. So here, this is us in G2. We have our exact copies of our 46 chromosomes. And what we're going to do is we're going to start going into cell division, or we're going into M phase. And M phase consists of both mitosis and cytokinesis. So during M phase, the DNA is going to coil up very tight. It is going to be in a different form called chromosomal form. And you can see right here that this is chromosomal form. You can actually see the individual chromosomes. We've taken that newspaper that's spread open, and we've twisted it so that it is going to be more organized when we are dividing, or when the cell divides, because we need to get half of the DNA, half of all of those 46 chromosomes onto one side, and then the identical copies of those chromosomes, we need to get those on the other side. And so we are going to organize these in these we're going to organize them by coiling them up, and they're going to be all coiled, and that's going to make it easier to separate. And you can see here that during M phase, each individual chromosome becomes more visible. So mitosis is the first part of M phase, and this is where the sister chromatids are split. And if you look here, the identical copies of the chromosomes go to either side. We can see that in this cell right here. This is a cell. These are pictures of an onion root tip. So the tip of an onion plant, the roots are growing into the ground. So there's lots of growth happening. And when growth happens, new cells are dividing. And so we can see lots of cell division here. And you can see how half of the DNA is on one side and how half of the DNA is on the other side. And so the chromosomes, 46 chromosomes went to one side. And over here, the exact copies went to the other side. And that is what happens during M phase. You can see how they're both still in one cell. There's two sets of DNA on either side of the cell, but the cell hasn't split yet. So that means that we have gone through mitosis, but we have not gone through cytokinesis yet. Cytokinesis is when the cell splits. So during cytokinesis, the cell actually divides into two, and the chromosomes begin to relax and uncoil again. They go back into that chromatin form that we saw at the beginning. And so right here, we started with one cell at the beginning of G1. We went through the entire cell cycle, and that one cell got bigger, and then it divided, and now we have two cells. And these two cells are both going into G1 phase, and they're going to go through the cell cycle again. And when we go into the G1 phase, we're going to need to be able to access that DNA again, because we're going to need to make more make more proteins and to get ready for S phase. And so it's important that during cytokinesis, when the cell is splitting, the DNA is also uncoiling, it's relaxing. So as it goes into G1, it's ready to start making proteins again. And I believe that's it. I hope that this helps, and let me know if you have any questions.